Okay. Now, today the presenter, Dean Liu, will bring us a very uh, uh, inspiring topic. In the past 3 to 5 years, uh, according to CDER's reports, novel drugs and biologics approval comprise more orphan indications than the regular indications. In other words, there are more orphan drugs and biologics receive approval than the non-regular drugs and biologics. But this group in Tsinghua University, we are friends, so I know that this research group, they go into and research what type of clinical evidence required for FDA to approve orphan drugs. So this is a very in, intriguing evidence, a very important support information for our regulatory affairs. Oftentimes, we like to know how FDA, what are the reviewing criteria by the FDA for orphan drugs. So thank you, Dean, to bring us this trendy, intriguing, and critical article to us. Thank you, Dean. The floor is yours.
Okay. Thank you, Dr. Guo, for your introduction. You almost finished my introduction part. I'm just kidding. Yeah, I'm Dean, and yes, I'll be nervous to be the first one. And this article is a clinical trial evidence supporting FDA approval of novel orphan drugs between 2017 and 2023. And I'm Dean. So yeah, this is how the papers looks like. So this is a table of contents and an introduction. Now there are more than 10,000 rare diseases worldwide and affect more than 350 million people. This is more than a combination of the cancer and acquired immunodeficiency patients. But among those, 95% lack of the approval treatment. So you can see this is a huge unmet medical need. To tackle this gap, to fill this gap, the US, they passed the law called the Orphan Drug Act on 1983 and to tackle this issue and also introduce the orphan drug product designation process and some financial incentives to support the development. And also they defined the rare disease. So the definitions is less than 200,000 individual. And I also look at different countries' definitions and their legislation. So after US legislation, other countries like EU, Japan, Australia, they all start their legislation. And the definitions of the rare disease is a little bit different between different countries. So for the EU, not countries, EU, it is less than 5 per 10,000 people. And Argentina is the same as the EU, and Japan is less than 50,000 patients, or less than 1 in 200 and 500 individuals. And in Australia is the same as EU, and the WHO is a bit different. It's less than 65 to 100 per 100,000 people. And Brazil is the same as the WHO. So this table is just information for you to know about the definition for the rare disease. But still, the FDA remains the global leader in the approval of orphan drugs with a significant number of approval compared with other countries. Okay, next page. So, but there is some concerns raised, like the critics in the evidence to support this kind of approval, like whether the evidence, maybe the evidence is a little bit lack of sufficient robustness, and which may lead to the uncertainty for patients. especially when they are like making their decisions to whether receive the treatment or not. They may misinterpret the result. However, the FDA say that because those drugs were in the specific circumstances, so there are some regulatory flexibility for those orphan drugs, but those substantial evidence were still in their statutory standard. So this study is to systematically assess the clinical trial evidence supporting the FDA approval of orphan drugs between 2017 and 2023, and also support the evidence standards and the regulatory flexibility. So what is regulatory flexibility? So the regulatory flexibility for rare disease shows it is 4 categories. The first one is quantity of the clinical evidence. Unlike the normal approval process, the sponsor only required 2 adequate and well-controlled trials, or I would say A and WC trials, or either they can conduct 1 single large multi-center trial and 1 AWC trial plus confirmatory evidence. So what is the confirmatory evidence? The confirmatory evidence refers to the additional data from 1 or more resource, like the clinical data, animal data, or the natural history of the rare disease, or either the data from the approval in the same pharmacological class, or animal data. And the second one is the flexibility in the type of the pivotal clinical trial, like the randomization, blindness, the type of control, like single arm or concurrent RCT, and the number of the patients or the duration of the trial, or either the endpoint can be flexible. The third one is the choice of the primary endpoint. Like the endpoint, maybe you can the sponsor can use the patient's feeling, functions, or either the validate surrogate endpoints, or like composite endpoint, and so on, to support their clinical evidence. And the last one is a statistical consideration, which is the type 1 error things. Okay. So then I will talk about the special regulatory programs in orphan drugs, not only in orphan drugs, but just regulatory programs. I believe most of you guys already finished your 511 class, so you may be very familiar with those terms. So I will just quick review about those terms. So for the fast track and the breakthrough therapy, they are all designation, and the feature is they have rolling review and some expedited development support. And for the accelerated approval, it is an approval pathway, and the feature is the sponsor can use the surrogate endpoint or the intermediate clinical endpoint that is likely to predict the clinical benefit. Those endpoints can be used in their clinical trial. And the last one is the priority review. It is a designation, and it can reduce the clock of the review time from 10 months to 6 months. Okay, so the method part is a little bit dry, but you still need to talk about that. So this study collect the approval orphan drugs, either biologics or drugs, from the CBER, CDER, and Orphan Drug database between January 2017 and December 2023 to assess the either the application type, either NDA, BLA, and dosage form, like the tablets, capsule, injection, and so on, and the route of administration, either it is intravenous, oral, and a product type like the small molecule antibody, and different therapeutic areas like anti-neoplastic or others. This study also assessed the special regulatory programs like those 4 programs I just mentioned. And this article also assessed the epidemiology of the orphan indications, which is the number of affected individuals and the prevalence rate for each orphan indications. They define the prevalence less than 6,600 individuals, or the prevalence rate less than 1 in 50,000 individual classified as ultra rare indications. Yeah. And for the statistic things, they studied analyzed the descriptive result to characterize first all the novel orphan drugs, second their orphan indications and the overall feature of their trials, and the third the characteristic of the clinical trial evidence at the level of the indications. And there are some more statistical tests for the data. This study also assessed the difference between the characteristic of orphan drugs or indications and the feature of their clinical evidence. And all the statistic tests were double-tailed and used 0.01 as their type 1 error rate. Okay, so the first part of the result is all the novel orphan drugs. The study enrolled 72 orphan drugs, and 45 of them, or 62.5%, were drugs, and 37.5 were biologics. For the dosage form, most of them were injection, 43.1%, followed by the tablets, 30.6%. For the route of administration, half of them were oral, 50%. And followed by intravenous. For the therapeutic area, over like 70%, 50 of 72 were in the anti-neoplastic and immunomodulating agents. And for the product type, about 45 of them, or 62.5% of them, were small molecule, and followed by the antibody. So in those 72 orphan drugs, were approved in 76 indications. So you can see 68 of the orphan drugs were approved in 1 indication, but 4 of them were approved in 2 indications. So this is 76 indications. And in those 76 indications, 49 of them, or 64.5%, were non-ultra rare indications, and 27 of them were ultra rare indications. And among those 76 indications, about 85% were using a special regulatory programs, either accelerated approval, priority review, breakthrough therapy, or fast track. And in those 30 accelerated approvals, 6 of them complete the post-marketing confirmatory trials, and 5 of them verified the benefit, and 1 was withdrawn. So this figure is just a quick summary for the dendrogram I just mentioned. So you can see red box over here. In those 65 special regulatory programs, 5 of them were used for programs, and 19 of them used 3 programs, and 19 of them using 3 programs, and 19 of them also using 2 programs, and 21 of them using 1 programs. And you can see this beautiful figure. Like the gray circle is project review, and the blue circle is accelerated approval, and the pink circle is fast track, and the breakthrough therapy is like a dark white, dark yellow circle. So yeah, just a quick summary of that. And the second part is the overall features of the pivotal clinical trials. So I will talk about the overall part first. So total in those 72 orphan drugs, or 76 indications, we have the study analyzed 87 pivotal trials, 87 pivotal trials, and in those trials only 49.4% were randomized, and most of them were open labeled and using the external control as their comparator, about 48.3%. And regarding to the primary endpoint, 72.4% were using surrogate endpoint as their primary endpoint. So if we dive into different categories, we can find a difference in the primary endpoint regarding to different dosage information. For the tablets and the injection and the capsule, most of them were using the surrogate endpoint as their primary endpoint, but like the powder and the suspension, they didn't observe this observation. Yeah. So there's a difference. And then for the different therapeutic area, in a type of blinding, you can see the anti-neoplastic and immunomodulating agents, drugs. Most of them were open labeled, 78.6% compared to 45.2% for the non-anti-neoplastic and immunomodulating agent. And in the accelerated approval drugs, most of them were not randomized. Only 22.6% were randomized. And most of them were open label and using external control as their competitor and using a surrogate endpoint as their primary endpoint. So this figure is visualized of the table I just mentioned. So just a quick summary. So for the anti-neoplastic and immunomodulating agents, most of them were open label. And in a serial approval and a non-serial approval, most of the serial approval drugs were not randomized and open label and using external control as their comparator and using the surrogate endpoints as their primary endpoint. So this is other result regarding to the patients in the clinical trial. So in those 87 pivotal trials, the median overall patients were 123, and 97 of them were in the intervention group. And the median weeks for the trials were 30 weeks. And only 41.4% of the trials conduct more than 1 year. The median overall complete rate were 92.5%. It's high, yeah. But there's a difference in different categories. In the therapeutic, in the anti-neoplastic and immunomodulating agents, the complete rates is lower in these categories compared to the non-anti-neoplastic areas. It's 95.2 versus 87, and it is significant. For epidemiology, it is quite reasonable. The ultra rare indications had fewer patients in their clinical trials, like 92 versus 100, 132, and 55 versus 108 in the intervention group. And in the serial approval, the median overall complete rate is lower in the serial approval drugs, which is 79.6 versus 94.5%. And it is significant, yeah. So the third part of the result is the characteristic of the clinical trial evidence aggregate at the level of indications. So we also look at the overall first. We have total 76 indications, and majority of them were using one adequate and well-controlled trial plus confirmatory evidence, which is 44.7%. It's majority is more than other two. Yeah, and for the trial design, most of them were using the concurrent RCT, randomized control trial, and using surrogate endpoint, 72.4% as their primary endpoint, and using the double two-side 0.5 p-value in 0.5 as their statistical considerations. And we dive into the different categories. There's, of course, is similar to the previous result. There's a difference in primary endpoint regarding to different dosage form. So the tablets and the injections, most of them were using surrogate endpoint as their primary endpoint, but the observation didn't observe in the other category, powder, capsules, and suspensions. And for the trial design, there are some difference regarding to the route of administrations. In the for example, in the oral, you can see most of the oral drugs were using single arm as their trial design. But in the intravenous and subcutaneous drugs, most of them are using the concurrent randomized control trial as for their clinical trial design. And then in the different therapeutic area, we can see the there's a difference in the pivotal trial for the anti-neoplastic immunomodulating agents and non-anti-neoplastic agents. So in the anti-neoplastic and immunomodulating agents, most of them were using one large multi-center trials for their clinical trials for approval. But in the non-anti-neoplastic drugs, most of them are using the one ANWC trial plus CE for their approval process. So it is quite different. And for the trial design, the anti-neoplastic drugs most of them were using single arm compared to the non-anti-neoplastic were using the concurrent RCT. And then we look at the accelerated approval drugs here. Most of the accelerated approval drugs were using the one large multi-center trial for their approval and using single arm as a trial design and the surrogate endpoint, using surrogate endpoints as their primary endpoint. And the similar result from the part 2, the number of patients is lower in the ultra rare indication compared to non-ultra rare, which is 100 versus 123. Okay, wait a minute. Okay, next page. So this figure is a quick summary of the table I just mentioned. So compared to anti-neoplastic and non-anti-neoplastic, in the anti-neoplastic drugs, most of them were using one large multi-center trial for their approval. And for the trial design, most of them are single-arm trial. And if we look at the accelerated approval drugs, most of them also using one large multi-center trial and using single-arm trial as a trial design, and using the surrogate endpoint as their primary endpoint. And all these data are significant difference. Yeah. Wait a minute. So in the discussions part, overview. In those 76 indications, 69.4% of the indications were in the anti-neoplastic and immunomodulating areas. And among those 85.6% of them were using the FDA special regulatory program approvals. And if you look at the 87 pivotal trials, only 49.4% randomized, 33.3% were blinded, and 48.3% were concurrent controlled, and only 13.8% were clinical endpoint driven. So this shows the lower evidence strength and greater uncertainty were acceptable for the orphan drugs when they get the FDA approval. And then wait a minute, yeah. And in those approval, only about 44.7% were using one acquired well-controlled trial plus confirmatory evidence to get their approval. So this shows the flexibility in the quantity of their clinical evidence. And the evidence these evidence are similar in one comparative study. And that study include about 25,000 clinical trials, and there are also significant difference observed in the rare disease trial and the non-rare disease trial. So this study consists with other studies. Yeah. And then, if we look into the different categories in non-anti-neoplastic and immunomodulating agents, 65.2% were using the ANWC trial plus confirmed the evidence, but only 4.3% of them were using one single large multi-center trial. Compared to anti-neoplastic and immunomodulating agents, which were using 35.8% were in the ANWC trial plus CE evidence, C confirmatory evidence, and however, most of them were using most of them, which is 49.1%, were using one single large multi-center trial. So these results show that the FDA should independently validate those results compared to different categories, because we can see the clinical trial design are different between two therapeutic areas. So the other discussions part regarding to the accelerated approval. The accelerated approvals were differ from the non-accelerated approvals regarding to a randomization, which is most of them were not randomized. Blinding, most of them were open labeled. Comparator, most of them were using external control, and of course the clinical endpoint. So this article talk about the surrogate endpoint. So although the surrogate endpoint can predict the clinical outcome, but it involves a significant uncertainty regarding to their relative to the real clinical outcomes. So when a patient using to decide whether to use the treatment, they may misinterpret the clinical outcome because of the surrogate endpoint. So which may lead to the drug may not actually improve the quality of life or extend survival for patients, either short-term or long-term. And to tackle this, the author suggests that regulatory agency should detailedly review the existing evidence for the efficacy of the surrogate endpoint to see whether the is there a chat? Okay. Goodbye, Dr. Pacifici. Okay, I'll keep going. And so the regulatory agency should carefully view the existing evidence for the efficacy of the surrogate endpoint to see whether the endpoint can truly predict the clinical outcome, and or the agencies can require the sponsor to provide the provide the confirmatory trials, which the result focus on the patient's truly clinical endpoint to truly predict the outcome. Yeah. And for the epidemiology, also have a little bit discussion of that. So the stratified analysis based on the epidemiology classified for rare indications revealed that the regulatory requirement and the flexibility didn't show the difference between the rare disease and the ultra rare disease. So expect the population, the patient, but the rest of them were non-difference. So this highlight the needs of the regulatory policymakers to make more urgent incentives to those fewer affect disease, like those very ultra rare indications. Yeah, and for a limitation, and also this article have some limitations. The first one is this article only analyzed the orphan drug designation rather than all orphan therapies. So this limit the comprehensiveness of the statistical finding. For example, we cannot use these results to fit in a medical device, or we cannot extrapolate it to other areas. Yeah, and the second limitation is practice surrounding market approval of rare diseases are highly complex. For example, there are not only CDER database, there are more database or resource for us to investigate. So the author mentioned some resource like CDER patient-focused drug development, model-informed product development, and some complex innovative trial design meeting programs. All these sources can be included into the investigation in the future. So the conclusion of this study is the study demonstrates the use of the special regulatory programs, diverse clinical trial design, and the flexibility quality of the clinical trial evidence. And we find the article found some difference in some categories, like the anti-neoplastic and immunomodulating agents, and the accelerated approval, and the rare disease and non-rare disease. There are some difference between those categories. So those findings highlight the need of the tailored regulatory strategy to ensure the consistency in the efficacy and the safety of the orphan drugs. So I think that is all my presentation. It's only 30 minutes. Yeah, thank you so much.
Thank you, Jean.